Hi, everyone. Thanks for the opportunity to share about what we're doing at Dimension Inc. and how we're using our biomaterials platform to create a new class of therapeutics. So we focus on regenerative opportunities. We are a regenerative therapeutics company. We design, develop, and manufacture these products to restore tissue and organ function. Just to kind of give you a sense of where we're at in our life stage, we have raised about $20 million to date. We have one strategic partnership in biopharma, and we're using our biomaterials platform as we continue to develop the platform to direct cells to restore function. The pipeline today is really focused on how we can unlock the potential of cell therapies. Our team is growing. We're based in Chicago. We're a spin-out of Northwestern University. One of my co-founders and our CTO, Adam, is in the audience, and joined by a growing team around us. We've got about 25 people today, a small GMP facility as well, and really also looking at building out the right set of people around us. So adding to our board and our advisors, and I've just included a few of them on this list as we're kind of really moving into this new wave of therapeutic opportunities. Probably not a new idea to many of you, but the central thesis about what we do is cells aren't enough. We need to be able to give cells the right environment to be able to function the way that we intend them to function. And this is, of course, how it is naturally in the body. And so what we're thinking about is how can you use materials to really kind of mimic that environment, to give the cells an opportunity to behave more like in the context of tissues rather than individual cells. How can we enhance that engraftment with the body? And when we see better engraftment, better integration, we then see better functionality as well. So on this slide, you see an image of one of our materials. We call it Fluffy. I'll show it in another moment as well. And it's a highly porous material. It has a great degree of surface area that cells really respond to. So the image on the top there is a scanning electron micrograph image of our materials, where this is what cells like. And so you see in the next image, cells, in fact, move into the materials, and you're able to see a very positive, kind of happy environment for those cells. What we believe we uniquely do is provide that full spectrum of being able to talk to a cell, if you will, at the subcellular level, but then all the way through to the macro level, and ultimately be able to provide something to a surgeon that they can implant or inject into their patients. And so we see a number of different ways that other companies and researchers are looking at delivering cells and promoting this type of cell attachment and ultimately the engraftment. Hydrogel approaches, which interact very well at the micro and nano scale, but maybe have a harder time with that rapid vascularization and engraftment due to that tight polymer network. And then you see more polymeric approaches, including newer 3D printed approaches that may integrate well in the soft tissue environment, but really don't talk to the cell and don't have that micro and nano nuanced structure that's really necessary for cell attachment and for differentiation. And so what you see in this image is the same product at different magnifications. So the product on the left is what the surgeon sees in the, on the right, what the surgeon will see in the OR, and then you kind of zoom in all the way to kind of the smallest area. And what you see is that we provide a lot of surface area. And again, that's where the cells really adhere, and we see some differentiation if we use stem cells to implant in our devices, as well as matrix deposition, and ultimately getting, again, the vascularization and rapid engraftment as well. So we've taken these materials and thought about, well, how can we actually apply them in the context? At our core, we're a platform company, but we all know any platform is only as good as the products that are developed on top of it. And so we've got a blended portfolio today of products that are internal assets as well as those that are partnered. And so here today, we're kind of sharing both sides of that opportunity, where we are ultimately looking for commercial partners for our internal assets, but we also see opportunities selectively to partner with, for example, companies that have a cell therapy asset that they're looking to enhance that engraftment or localization or the cell viability, where we may be able to co-develop an ultimate solution for patients. So our first product is, and I'll talk about this in a moment, is actually a medical device. It is an acellular product really designed to de-risk the platform and show how our materials alone act on endogenous cells. But where we're really moving towards is how we can combine cells in our materials, for example, in the case of type 1 diabetes, to provide an encapsulated device to be able to deliver insulin-producing cells. And we'd have one undisclosed partnership as well that's earlier in discovery. So we're a therapeutics company talking about devices. So why did we start with a device? And the reason is really very simple. We wanted to show that our materials alone can control cell fate and ultimate cell behavior. So this is a product that doesn't use any cells or growth factors. It really, again, acts on the endogenous cells within the body. It is an implant. It's the same one you saw a couple slides ago, and used by plastic surgeons or oral maxillofacial surgeons for facial bone reconstruction. So think a defect in the patient's face due to trauma or maybe a congenital malformation. It may be for aesthetics purposes. It might be that you need a tooth implant and need to enhance your jaw. In the case of our early animal models, we used a jaw defect, for example, in the model you see here, where this is a large pig, where we took a defect in its jaw, and we're able to regenerate that bone through the use of our technology. And over a period of 12 weeks, we saw, again, healthy bone, similar to native tissue, to the surrounding native tissue, and ultimately restore the contour of that. And we believe that it's largely due to the fact that we do see this very rapid vascularization and tissue integration. It's also a very surgically friendly material, and so it has a little flexibility to it. Surgeons get it in the OR. They're getting excited about using it. We actually just implanted this in our 10th patient today, so this is very new in terms of our commercial launch here. But what we're seeing is that surgeons can cut it and shape it to fit the patient's defect, and then over time it remodels into natural bone. One of the things we're able to do is to tailor our materials for specific indications. And so in the case of facial reconstruction, one of the problems is volume preservation. Basically, the materials that are often used today, they'll almost absorb faster than the bone forms. And so you got to match that ability to, for resorption and bone formation. And so this is a canine model for a tooth socket, a tooth extraction, where it's really important for us to maintain the volume of that surrounding tissue as the bone remodels and grows back. And so we see bone grow from the inside out and the outside in, and get this, again, robust, healthy bone tissue in a matter of weeks. So again, we're a therapeutics company. Why did we look at a device? We wanted to de-risk the regulatory pathway. We know these are combination products that are going to involve both CBER and CDRH, and so we wanted to get a read from CDRH and how our materials and our 3D printing process were going to be assessed. And so we received FDA clearance from the FDA at the end of the year, literally December 30th. We've set up our GMP facility in Chicago for small batches. We're now delivering our product to patients through surgeons as part of a limited launch that will go through early part of next year. Where we're moving, and really where the vision of the company is how can we create these tissue-like environments to enable localized delivery of cells. And so you see here what we call our fluffy sheet, and think of it as like a patch that could be laid, for example, on a soft organ. And you can see in the video, which I'm glad is working, that it's very malleable. So surgeons can actually, manipulate it within their hands, and it maintains its structural integrity. So again, going back to that slide where the length scales are important, we talk to the cell, but we also create a product that has the robust enough integrity for a surgeon to manipulate it and for cells to be added to it, even at the operative site. What you also see here is a great example of why cells like our materials. So on the top image, you see what would be more of a traditional printed polymer approach. Then we use that same material through our proprietary process, and you see what's on the bottom images. And as we create that micro and nano porosity and topography, that's where the cell adhesion sites become available, and you see that cells, in fact, do proliferate within our materials. once these are implanted, we do see that rapid vascularization and engraftment. So this is a 6-month rat study looking at a tracheal defect. So in this case, the product was actually printed into a coil that was essentially wrapped around the tracheal defect in the animal. And over a period of that 6 months, we saw that nice integration and maintained the patency of the trachea. It also allowed for that defect to be sealed. And so you see the lack of fibrosity, just a nice, healthy integration. And again, these types of materials you can imagine adding cells to for additional applications. So just to kind of give you a sense of imagining what types of possibilities. I mentioned earlier, these are flexible grafts, and so we can print them in a range of sizes and shapes. But one of the things that we hear most often is, can we place our cells on a sheet? Can we seed our cells onto a sheet that can be placed on an organ? In this particular case, we're looking at a cardiovascular an infarcted model where we're looking at creating an ability to restore the conductivity of tissue that has been infarcted. And so this is actually using a material that is primarily graphene. So we can print different materials. Graphene is an electroconductive material, and so printing this into a sheet, placing that on the cardiac muscle, and being able to show that we can restore electrical function across that infarcted tissue. What you see on the left side of this image is that same patch, but being seeded with cells. So we have not looked at that cell response in an in vivo model, but in an in vitro model, we are able to see that as we tailor the micro and nano structures within our device, we get varying degrees of cell response. And again, something we can tailor to mimic that natural tissue-like environment that we're looking to create. So we can use these kinds of specialized systems to develop new tissue therapeutic products. So we believe this is kind of the next wave of types of therapeutic solutions where we're combining materials and cells to be able to restore function. So internally, we're in the late discovery stage around type 1 diabetes application, where we're creating a cell encapsulation device that is designed to overcome some of the challenges that we've seen with others and getting that nice engraftment and that healthy, harmonious integration with the surrounding tissue. And so, again, early stages of development here, but we already see in some of our in vivo studies that we get that integration and vascularization that we're looking for, which should translate then to improved viability of the cells and ultimately of the graft over time. So we're really excited about what we're building with our platform at Dimension, and really looking at different opportunities. Why we're here at this event is really to continue to talk with others about what we're doing in our own internal pipeline, be able to have those initial conversations with groups that could potentially serve as our commercial partners as we advance those assets, but then also looking for selective joint development opportunities. We have one of those today, as I mentioned, that we were able to license the use of our technology in a particular area and then provide joint development for those products. And so looking to engage with companies maybe who are having challenges or see next generation versions of their products to be able to increase that therapeutic potency. So I'll be around for the remaining hour or so of this conference. Happy to answer any questions, but please also feel free to reach out. We are actively looking to get feedback and to explore new synergies as well. So thanks all for your time today.